Hello, my bacterium is Corynebacterium diptheriae. This is a scanning electron micrograph of the bacteria here. Corynebacterium diptheriae is a bacterium that causes diphtheria. Though diphtheria can be expressed as a skin disease, it's mostly commonly expressed as an upper respiratory bacterial infection. Diphtheria comes from the Greek word for leather, based on the manifestation of a tough grayish pseudomembrane in the back of the throat that's made up of fibrin, dead tissue, and bacterial cells. Over time, it can grow completely to block the passage of air to and from the lungs, so it's a big uh, identifying factor of diphtheria. Symptoms primarily include a sore throat, fever, general discomfort that is commonly attributed to the presence of an infection, and the swelling of the neck from enlarged lymph nodes. Until 1935, diphtheria was the leading cause of infection of children in the U.S. And then another important fact is that it was the first disease for which a toxic cause was identified. So these pictures show that pseudomembrane there. The culprit is Corynebacterium diptheriae. It's a bacterium that's gram-positive, it's a facultative anaerobe, and it's commonly found in soil. As far as morphology goes, it's a rod-shaped bacillus, and it's also described as pleomorphic due to its ability to sometimes become club-shaped when it is dividing. It does not possess the capsule, but it does possess the gram-positive, characterizing thick peptidoglycan cell wall. This bacterium is also acid-fast, which is similar to the tuberculosis-causing mycobacterium, meaning it contains mycolic acid in its cell wall, which might make it harder to gram-stain and penetrate with antibiotics. C. diphtheria is non-motile. It lacks flagella, but it does possess pili that aid in the bacterium's attachment to the mucosa of the respiratory tract. This bacterium is ultimately characterized by its protein-altering toxin, which is known as the diphtheria toxin. This toxin lies inactivated in the bacterium until a bacteriophage comes along, infects the bacterial cell, activates the toxin, and thus turns this once harmless soil bacteria into a deadly pathogen. And we'll discuss that further down the line here. Transmission and frequency of infection. Diphtheria is most commonly spread via droplet transmission. So someone infected with a bacterium that coughs or sneezes can expel the bacterium into the air, where someone else can breathe it in and become infected. The droplets are extremely resistant to drying, so it just gives it more virulence there. The preferred portal of entry and exit is the respiratory tract. So once transmitted into the nasal passage, C. diphtheria hunkers down and begins to colonize in the surrounding mucous membrane, usually only after the toxin's activated. And then as time passes, the colonization can spread down to the throat and further along the respiratory tract until it reaches the bloodstream, where things could get very serious if the toxin's been activated. Respiratory cases of diphtheria have been known to also arise from contact with cutaneous diphtheria, which is characterized by lesions on the skin. You can also find that pseudomembrane that forms on the skin. Here's that pseudomembrane here with this child coughing. According to the CDC, there are about 200,000 reported cases of diphtheria in 1921, which resulted in approximately 1,500 deaths. But the introduction of vaccines has reduced that, and now today, annually, there's about maybe 5 cases reported. Outside of the U.S., however, the World Health Organization gets about 7,000 cases of the disease, or got about 7,000 cases of the disease in 2016 alone. And that could be attributed probably to the lack of access to vaccinations in a lot of developing countries. CDC also states that the treatment for diphtheria was discovered, or after it was discovered, the fatality rate of the disease was 5 to 10%, with an increased rate of 20% in younger individuals. Mechanisms of pathogenicity. The important thing to note here is the diphtheria toxin. So the bacterium doesn't invade tissues, the toxin does. So, when a bacteriophage comes along and lyses that bacteria, it also activates the toxin, which then leaves that bacteria, finds a host cell bacteria. It's an AB exotoxin, which means it's going to leave the bacteria cell, find a host receptor cell, find the receptor. The B component of the toxin attaches to the receptor of the cell. It becomes engulfed by the cell, Once inside, the A component, which is an enzyme, releases and starts messing with all the mechanisms inside. And pretty much the big thing it does is here. Oh, right here. Sorry. It hmm, It inhibits protein synthesis by means of catalyzing ADP ribosylation of the elongation factor 2, which is a huge deal as far as translation goes in protein synthesis. And then as the toxin spreads from host cell to host cell, it can eventually make its way into the blood, which is going to disrupt the functionality of the heart and other major organs, which is a no-go. And only zero point 0.01 milligrams of this extremely virulent toxin can result in death. So let's see, diagnosis and detection. The diagnosis of this can be done using a few laboratory methods, including gram staining, something called Loeffler's medium, and tellurite blood agar for isolation of the bacterium. The primary purpose is to isolate it to determine whether or not it's toxic. So a gram stain can be performed to show that it's gram positive, as seen here. And then the back of the throat of someone infected is swabbed with a sterile cotton swab. It is then grown on this blood agar medium until it is isolated. And then the next big test is the ileic immunodiffusion test, which is a lot like the Kirby disc diffusion test, where they lay a strip covered in the antitoxin over the media of the isolated bacterium, and if it moves away from the strip, that shows that that toxin is active and that that person is infected with a active toxin. And it's hard to diagnose without these accompanying laboratory tests. So even if you see the pseudomembrane, further tests need to be done to show that it is diphtheria. 1923, according to Microbe Wiki, Alexander Glennie, Barbara Hopkins, and Gaston Ramon produced the vaccine for diphtheria that is known as the toxoid vaccine. So it is a treatment for infected individuals, but it involves you need when you're infected to do antitoxins and antibiotics as well, because antibiotics are not going to kill the toxin, they'll kill the bacteria. So you need accompanying antitoxin to halt further production of the toxin in the body. Common common antibiotics are erythromycin and penicillin, and the most common vaccine is given as a booster throughout life called Tdap. It's for tetanus, diphtheria, and pertussis, which is whooping cough. So this is done throughout life a few times when you're young, and then as you get older every 10 years, so that you can ensure that you are still immune to these diseases. That is Corynebacterium. I'm not sure if I have more time here, so I'm just going to stop. Thank you for watching.